Right. Hello everyone. Today I'm going to talk about how I use nanowells to investigate the role of paracrine signaling in the release of neutrophil extracellular traps. So we all have flu had cut on our fingers before. Then what's in our body protect us from the outside invaders, from things like bacteria, fungi? The answer is the immune systems. The immune cells can form an army to fight against the invaders. Today, I want to talk about my favorite immune cells, the neutrophils. Neutrophils are one of the most abundant white blood cells. They are the first cells to go to the site of infection. So people who don't have functions in neutrophils or don't have the right amount of them will suffer a lot from infections. Neutrophils could be activated by lots of external stimuli, from things like the bacteria, the damage-associated molecular patterns. Then what activated neutrophils could do during inflammatory response? They can actually kill microbes to prevent further dissemination through phagocytosis, releasing antimicrobials and web-like structures, neutrophil extracellular traps, and we call them nets. It could also send alarm signals to other immune cells to propagate the inflammatory response. Today, I'm particularly interested in the production of nets, a process also known as mitosis. And nets are primarily composed of DNA and histones. They could trap and kill microbes. So clearly, nets are good for us. They protect us from lots of threats. However, the excessive netosis and the impaired clearance of nets could cause lots of inflammatory disorders. For example, the nets could provide stimuli to promote the thrombus formation by promoting red blood cells and platelets adhesion. It could also induce organ damage in autoimmune conditions. So overall, NET is a double-edged sword of immunity. So we want to inhibit the excessive netosis, but we still don't know why and how that happens. So some studies have shown that neutrophils could release inflammatory mediators during netosis. They can release things like the reactive oxygen species, the ROS, and the interleukin-8, which is also a well-known netosis inducer. Since neutrophils have the receptors that can bind these mediators, we think probably the activated neutrophils could trigger netosis of other neutrophils. So my goal here is to investigate their signaling pathway, what kind of paracrine signaling they are using, and how it works. So to limit the paracrine signaling, the extracellular communication, we used nanowells. So to fabricate it, we used PECDA 250 hydrogels and a laser patterning system. So first, we will load a small volume prepolymer on a glass slide, and then use a PDMS cover to make it spread evenly. Then we need to use a UV laser to photopolymerize it. After that, we will use the ethanol to rinse out the uncured stuff, and in step 3, we will assemble the plate frame with the glass slide. And as shown in this picture here, we can see that the nanowell unit size is around 80 times 80 micrometers, and the wall height range 26 to 35 micrometers. For the netosis quantification, we usually need to stain the cells. So for my experiments, I stain cells with the Hokist, and for cells can netose, they will go through the first 3 stages. So at the beginning, of course, they are healthy cells. They look very bright under the microscope. Then the cells exhibited the DNA decondensation. And at that time, the cells still have the intact membrane, so the fluorescent intensity will still be very high. But after the breakdown of the cell membrane, the DNA will go outside of cell membrane, and the fluorescent intensity will drop dramatically. In the mixture, we could also see something like the DNA fragments and some dead cells. And for dead cells, they will look very bright. For my experiments, I use a kind of neutrophil-like cell. I call it DHL60 cell. And in the control group here, so without any NETosis inducers, after staying with the Hawkist, they look very bright and very rounded in very good shape. But for the treated group, like the cells treated with the NETosis inducer, we could see some web-like or cotton-like structures. Those are the nets secreted by the activated cells. And if you follow the yellow arrows here, we can also see some DNA fragments. We used automatic cell counting for the NETosis quantification, and I needed to highlight, mark the intact cells in red based on the fluorescent intensity, area, and the circularity. Here's the example. For my experiments, I used L-lysine calcium salt as the only NETosis inducer. In the control group here, we can see almost all cells have been marked in red, and those will be treated as intact cells. But for a picture on the right, those are the group treated with the L-lysine. So some of them still have the characteristics of the intact cells. There are some intact cells there, because not all cells can NETose. And we can see some examples of, if you follow the yellow arrows here, you can see some examples of those activated cells that secreted nets. They cannot be marked in red. And to get a percentage of nets, I basically need to take two images. I will take my first image immediately after adding the L-lysine, the inducer here. Then the total number of cells will be the number of intact cells, because the cells cannot respond that fast. And after around 4 hours incubation, I will take my second image. And in the second image, I will count the number of intact cells again. In that case, the difference between those two images will be the number of cells that secreted nets. And if you follow the formula at the bottom, you will calculate the percentage of nets. And here are some results. Figure A here, we can see that the percentage of nets increased over time. And we usually wait 4.5 hours, and that's enough for the most cells to NETose. For the cells, we also conducted experiments inside the regular well plates and the nanowells at the same time. And we used the same concentration of the L-lysine, the NETosis inducer here. So when the cell density was high, the nest level inside the regular plates was higher than the nest level inside the nanowells. But when the cell density was low enough, the difference is not that much. We also tested on different cell densities. For the untreated cells, those are the cells in the control groups. So no matter what the density of this cell is, they still have around 10% of nets. And for other treated cells, we can see that the higher density of cells, the higher level of nets. And it also showed a strong linear correlation between the cell density and the percentage of nets. We then want to see, we employed experiments to see whether the activated neutrophils could directly trigger NETosis of naive cells. And here's the experimental design. We first treat cell batch 1 with the L-lysine for around 25 minutes. Then we wash out the L-lysine and replace it with fresh medium. Then we added the cell batch 2. So that means cell batch 1 and cell batch 2 will be cultured together inside the incubator for another 4.5 hours. And in the end, we only analyze the cell batch 2. That means we only calculate the percentage nets of cell batch 2. That's because we only care about the naive cells. And here's the results. The x-axis shows you the density of the treated cells. That's the cell batch 1. And the y-axis shows the net level of the untreated cells. That's cell batch 2. So we can see that the percentage of nets depends on the density of the treated cells. Once the density reached a threshold level, the nets of untreated cells, the percentage nets of untreated cells didn't change a lot. Here in conclusion, we provided a high-throughput system for the investigation of paracrine signaling. We can conduct experiments, like 1,000 experiments in parallel inside the nanowells. And we found that these nanowells did limit cell-cell communication in comparison to the regular place. And to verify the paracrine signaling, we tested on different cell densities, and the activated neutrophils could directly trigger netosis of the naive cells. In the end, I want to thank my supervisor and all the colleagues here and the funding agencies. Thank you all. Now I'm happy to take any questions and comments.
Thank you for that great talk. I'm wondering if you'd taken just the cultured cell media and used that to just highlight the secretion is giving rise to these effects that you're seeing as a consequence of cell density, as opposed to just cell-cell contact is giving rise to these activation states that you're seeing?
Yeah, I mean, that's a really good comment. I mean, we thought about what triggered the netosis of the naive cells. It could be, as you said, the cell-cell contact, or it could be cytokine released by the activated cells, like soluble molecules, or it could be some proteins that stick to the nest, because nest could be very sticky. And we still don't know yet, but that would be our next stage to test like supernatant and to test different proteins and test cell-cell contact to see what we can get.
Yeah, that'd be terrific. Or even DNase treatment to exclude.
Yeah, DNA treatment. Yeah. Some studies also showing that if you like, they used the mitochondrial DNA extraction kit, and they proved that mitochondrial DNA could directly trigger netosis of human neutrophils. So DNA might help, but we wonder whether it is the only thing that triggers netosis of naive cells.
Well, what a fantastic high throughput tool you're developing. Congratulations, terrific.
Thank you.
I was wondering if you could speak to the clinical applications of your research.
Okay, thanks for the comments. I guess because we want to inhibit the excessive netosis, because excessive netosis could cause like lots of inflammatory disorders if we know the mechanism behind it, like what triggered the excessive netosis. We probably could find a way, probably like add medicine or drug tests to how to inhibit. But the first thing we need to know is the mechanism behind it. Then we know where to start. Thank you.